How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, are all made of DNA. We're all made of the same chemical DNA. We're all made of DNA. We're all made of the same.
Hi, you're listening to DNA Today, a podcast and radio show where we discover new advances in the world of genetics. From genetic technology like CRISPR to rare diseases to new research, we have you covered. For a decade, DNA Today has brought you the voices of leaders in genetics. I'm Dineen. I'm a certified genetic counselor and your host. TrackGene has designed a genetics electronic health record. Here's what it features: pedigrees, demographic data, genetics information, risk tools, and sophisticated reporting, all within a clinician-designed workflow. It integrates with other clinical genetic software, databases, and hospital information systems to maintain accurate patient records. Go check it out at trakgene.com. Again, that's TrackGene without the C.com. And be sure to check out episodes 208 and 210 of DNA Today, which features TrackGene. When I began working as a genetic counselor, I was mostly starting from scratch because the private practice I joined hadn't had a genetic counselor in around 2 years. So when I started, I met with a bunch of different labs and asked them about their tests so I could make a decision on who I'd be ordering from and what scenarios, and just gathering all that information. But I have to say, it's a brand new GC, which I don't think I can quite claim the title anymore because I graduated 2 years ago. But at the time, this was really hard to figure out. It was hard to figure out what the important aspects were. And so I figured it would be really good to dive into this aspect of genetic counseling and genetic testing, rather, more so in this episode, and focusing on the quality side of that. So to do this, I needed some guests, so I figured let's bring on two experts. I'm joined by two experts from Blueprint Genetics, Dr. Kirsty Wells, who is a senior geneticist at Blueprint, and Rachel Goldberg, a genetic counselor. So thank you both so much for coming on and having a more unique episode this round.
Yeah,
thanks for having us. Yeah, good to be here.
Why don't we start out with really the question that we're going to be answering throughout this, which is, how do you define a quality genetic test?
Yeah, I can go ahead and start on that one. You know, I feel like there are so many ways to answer that, and it truly depends on perspective, right? So I started out in clinic and then went over to industry, and I think when I think of quality, it's kind of this three-pronged relationship between patients, providers, and the lab. And it's about trust, right? It's trust that you are offering a test that's going to be looking in all the appropriate places to find, give the best chance of finding an answer. It's about giving options for people of different socioeconomic statuses. It's about making sure you're doing right by the patient and their information. And it's doing right by the providers, right? Making sure we're educating them and transparent about what they've got to offer. So it kind of depends on the lens that you're looking through.
Yeah, absolutely. Completely agree with what Rachel says, that there are so many different and all correct answers to this question. As a geneticist, I'm kind of into the side of the analysis and the reporting, the more technical side. So what I would say is that a quality genetic test is one that performs well enough technically to give you the best possible chance of finding a molecular diagnosis for the patient, and that actually produces results that are meaningful and that you can actually trust. So in practice, this means a number of things, like good coverage, especially of difficult to sequence genes that might be relevant, a broad scope of the test, so what types of variants it detects and what genes are included, high-level interpretation, and a meaningful report which is easy to understand. So there's lots of different elements there which all come together to make a quality test. So it's not just one thing in isolation.
Yeah, you highlighted, both of you, Rachel and Kirsty, so many different aspects of a genetic test that is really like our dream genetic test. It would have every single aspect that you talked about. But in practice, it's often we're choosing, well, what's the most important? What's our maybe top three? Something along those lines. And And before we dive into all of these questions I have as we explore deeper aspects of so many of what you just gave us an overview for, I wanted to give a little context that Blueprint Genetics was founded the same year as the show started, actually, so 2012. So this is a decade ago. A decade is such a long time in genetics. This is just, in some fields, it's like, oh, that's nothing. But in genetics, that's a really long time. So what has changed in the pursuit of offering quality genetic testing if you're comparing when Blueprint started to where you guys are now? I'm sure there's some aspects that are the same, but with increase of technology and the advancement of technology, I'm sure some of this has probably changed.
Yeah, we've come so far in the past 10 years. I mean, again, on the technical side, I think we've come in the past 10 years from kind of genes, tests looking at single genes or just a single type of variant, to more complex tests that look at lots of different genes all at once and lots of different variant types. So now using NGS, we can look at lots of genes and lots of different variant types, small copy number variants, deep intronic variants, mitochondrial variants. So all of that in one test, I think, was not possible 10 years ago. So thanks to that, we have an even better chance now of finding a diagnosis for the patient the first time with the first test.
I think it's kind of funny, just even when we think about, Kira coming out of school and what you learn in school versus what you learn now and in practice. I mean, just like Kirsty said, deep intronics were not something that we were really worried about. And it's still not something that's standardized to include in all genetic testing out there, especially when we have identifiable pathogenic variants in deep intronic areas. So just that concept alone is huge. And just thinking about how much there is to talk about and include, like genetic counseling in itself, those kind of visits are so long and there's so much in there. And thinking about where we are now and how much we have to fit into our short amount of time with a patient, how much are we going to try to fit in, another decade from now, or maybe it will come full circle and it's all going to become sort of something that's in one test versus now I feel like we're still in that stage of, well, how targeted do you want to be? How much time do you have? Cost is always a factor. And I feel like it's become more complex over time, and it's hard for me to tell if it's going to become even more complex or maybe things will kind of start to come together a bit more.
Yes, I think Laura Hersher has a great quote that she has in her Twitter. You're nodding, you know where I'm going to go with this. That genetics, it's a lot more complicated than we thought. And the more we learn about genetics, the more we're like, whoa, how does this work? And I mean, that's why we still do this show 10 years later, and you guys are still running, because there's just so much that we're learning and uncovering, and the amount that we're able to do is even more so. Kirsty, you mentioned NGS, so next-gen sequencing, and an important aspect of this is to get high-quality data from the sequencing, and part of that is the depth of sequencing. Can you define what depth of sequencing means, and is there a requirement for it to be considered high quality? Is there not? Like, is there a standard there? Because that's something like I don't know the answer to.
Yeah, and we kind of throw around these terms, sequencing depth and sequencing coverage, and I think it can sometimes be a bit unclear what we mean by that, but these are really important concepts when it comes to quality. So what we mean by depth is the number of times that a particular nucleotide in the test has been sequenced. Because in NGS, each nucleotide is read multiple times to give you a variant call, and the more times the better, because then the more times a nucleotide is read, then that increases accuracy and confidence in the data. Now, ideally, we want to see a minimum depth of 20x, so that's a nucleotide sequence 20 times. To kind of be sure that we have a reliable variant call. But what I would say is that it's not just about depth, it's also about coverage. And what that means is how many of the nucleotides on the test are covered to that minimal level of 20x, and we express that as a percentage. And on average, we want to capture and sequence well over 99% of the nucleotides on the test, to kind of be sure that we're covering all of the genes, all of the kind of different regions where you might see a relevant variant, to kind of a high enough level. So what we like to say is it's like a pizza, depth and coverage. So you want a good, thick, kind of cheesy, tomatoey pizza topping on your pizza, and you want it all the way across the pizza. You don't want it to be patchy. So good depth and good coverage is important in your pizza, but also in your genetic test as well.
I love that analogy. It's making me hungry for New York pizza.
Me too.
Me too. Isn't it funny how, I coming out of GC school didn't really know that. I, in clinic, kind of had concepts of what that meant, but it wasn't until I was in industry and got to work with people like Kirsty and understand what really depth and coverage means. And to go a step beyond that is, what does it mean if it's just slightly different between labs? What does that mean in terms of how much information is not looked at? And even though it could be different percentages of coverage varying by just 1% or so, that could equate to many genes that are not looked at appropriately. And then to go on, what does that mean for a patient? What does that mean for a report and the security that a lab could give falsely if you're not doing the same amount of depth and coverage as another lab?
Fantastic point, because as we're saying, like, if it's like 98% versus 99%, people are like, oh, like you hear that in other contexts. You're like, right. Okay, like you got a grade or something, right? You got a 98 on a test versus a 99. It doesn't really make a difference. But here, that's representing such a large number. So that 1 or 2%, as Rachel's pointing out, this could be multiple genes that are not being properly sequenced to that depth that we want to see, and that coverage meaning that they're not even included there. Kirsty, you have something to add there?
Yeah, I was just thinking that those kind of tiny differences in coverage, well, the perceived tiny differences, but they actually translate to large differences. They become really important when you start thinking about genes that are kind of known to be difficult to sequence but are clinically relevant. So those are the ones that, with these kind of differences in coverage, those are the ones that you're probably not covering as well. So for example, PKD1, a gene associated with autosomal dominant polycystic kidney disease, is known to be really difficult to sequence because of its genetic content. It's very GC rich. There are multiple pseudogenes. It's a very, very difficult gene. So if it's not covered well, then you could miss potentially relevant disease-causing variants in that gene. So if you have a patient with polycystic kidney disease and that gene is not well covered, there kind of becomes less point in testing in the first place because your main kind of relevant gene is not well covered.
It almost feels more like screening at that point than testing if you're missing a chunk, because then that feels more like carrier screening. Like, as a prenatal genetic counselor, that's kind of what I'm relating that to.
Have you heard of TrackGene? TrackGene is a clinical genetic software solution used by over 1,000 genetic experts around the world. You can customize the front page so it's streamlined to your specific workflow. The intuitive patient information entry page makes data entry efficient and user-friendly. Pedigrees are also easy to draw and document. Here's another vital feature. It supports HL7 integration to be used with other clinical genetic software, databases, and hospital information systems. So you can build custom reports with the simple drag and drop report builder. This has an interface with data visualization tools such as those from Microsoft to make it easy workflow. And there's more features on the way, all designed with you in mind as the genetics expert. TrackGene has an experienced team who's been working in the clinical genetic industry for, get this, over 15 years. You can request a demo for free. Go to trackgene.com. Again, that's TRAK gene, so track without the C.com. And be sure to check out episodes 208 and 210 of DNA Today, which features TrackGene.
So, we're kind of going through the process here. Once quality data is obtained, and the lab has that, the next step is to analyze it, as we've started talking about here, of looking at the variants and being able to classify them. You know, I feel like Kirsty's going to nerd out with us for a second.
Yeah.
What are the AI programs?
I can't wait.
What are the AI programs, tools, techniques that either you utilize, Blueprint does, your colleagues do? What are some of your faves?
Well, I mean, it's kind of good human brain power, really, is my favorite tool. So when it comes to interpretation, it involves really being a detective and gathering evidence from loads of different sources, weighing it up, and to make an assessment on how likely that variant is to be disease causing and whether it's relevant to the patient. And there are lots of sources and tools that we use to do that. But it takes a kind of geeky human brain to sort of bring it all together. So, I mean, the sort of thing we would look at, we look at the frequency of the variant. So is it rare enough to plausibly cause rare genetic disease? And we have databases to help us with that, like Nomad. We consider the inheritance pattern. We take a look at the predicted consequence of the variant. So is it a truncating variant, which could cause a loss of protein function? We have various kind of in silico tools that we use to make predictions about the variant. I would say the most important thing, though, is kind of really going into the peer-reviewed literature, doing a deep literature review, reading papers, reading about patients that have been reported to have the same variant that you're looking at if there are any experimental studies that could be relevant. So properly kind of interrogating and understanding the medical literature is a really key part of quality variant interpretation. And you mentioned AI tools, and I think this is a very hot topic in kind of variant interpretation now. And I would say they can definitely facilitate the interpretation, so they can kind of help to prioritize variants to make the geneticist's work kind of easier. But what I would say is that a lot of the time it still takes a human brain to catch those difficult diagnoses, to deal with the tricky variants. Variant interpretation can be quite complex and nuanced. So I think what I'm trying to say is that human supported by machine in variant interpretation is I think what leads to the best quality result at the moment. Sorry, waffly, long and geeky answer. Yes,
well, I knew it was going to be, right? That's fantastic. And as you said, like, you need a human brain that is geeky to be able to do this and use the tools that you've mentioned and the methods and different process to go through this. One thing that I was wondering about before this interview is that obviously you guys use like a reference database to say, okay, here's a database of what we think to be normal human changes here, because my DNA is going to be different than each of yours. That's why we all have, for instance, different color hair, right? That's something that's very apparent for the three of us. So that was just a lucky thing to pull out there.
Right.
Very easy. We can see this, right? I don't know if people dyed their hair. Don't say that. That's going to ruin my point. But with that, neither of them are bad, right? It's just different. So human difference there. But that our variants are normal. So that would be in a database. Say like, oh yes, this one is, dirty blonde, this one's black hair, this one's ginger hair, great. So how large do these reference databases, I don't know what word I want to use, I guess like need to be in order to be useful? Because if you only have like 10 people in this database, like when we started out, we only had like 10 genome sequence, that's not enough to say, well, no, none of these 10 people have this variant, must be pathogenic. So at what point do we say, all right, this is substantial evidence to that?
Yeah, tough one.
It's not an easy answer, right?
Yeah, there is no easy answer. There's no cutoff. I mean, obviously, the more individuals you have in a database, the better, because then that gives you a better chance of being able to exclude something. I mean, with databases, I would say it's not just about the size, it's about the diversity of them as well. And I think a key topic in variant interpretation is that a lot of these databases we use contain sequencing data from individuals of European origin. So that makes interpretation of variants from other populations more complicated. So large databases are great, but kind of diverse databases are even better.
Yeah, that's definitely a point we highlight a lot on the show, because it's just one that I think has been talked about more in the last couple years, but we're still not getting to a point where I feel like the field is really addressing it head on. I think it's being addressed, but it's not as fast as I would like to see.
Yeah.
Certainly, like when people do testing and they're not of European descent, or they have a mix of ancestry, their genetic testing is not going to be as accurate as someone that has just direct European descent. And that's just a huge problem.
Yeah.
Right, or it might take more time. I just feel like time is something that comes into play with so many things that we're talking about. I mean, with Kirsty talking about how much time goes into for human brain power to really interpret these variants, you know, when we're thinking of quality, how do we mesh it all together to be able to provide that quality with the time it takes with the need of the patient, right? If this needs to be something that, we need to have something rather quickly because of patient health, or perhaps this is a diagnostic odyssey and a couple weeks is not something that's going to change the big picture. But I always found it funny when I learned how immense actually the data that comes out of here. You know, if you ask any clinician out there, how many variants do you think are seen when someone does a test? Let's say a panel of a couple hundred genes. How many do you think there are?
Oh, that's a great question.
And Kirsty,
I'm on that side, right? So like I would
right.
probably say like, I don't know, variants, I mean hundreds, I would think. And then having to sort through all that. It's just like crazy. I'm like, wow, that's a job that would really, really stress me out.
It could, right? And I love Kirsty highlighting like all the fact that AI is not something that is a bad thing, right? But it is not something that really can stand alone without people looking, just because of all of these nuances, our hair color differences, our health differences, different parts of the world where we come from. It really takes a lot, because something that might perhaps be passed over if you're just using AI could be something that is really identified by a human brain, like a Kirsty.
Yes, exactly. And Rachel, you brought up, the diagnostic odyssey, which is really important, especially when we're looking at the rare disease community, because people are often on this odyssey for years before they get an accurate diagnosis. And part of when healthcare providers are ordering genetic testing, they often provide, if we're talking about kind of this rare disease pediatric setting, they're often providing phenotypic data to go with that. So we can help the people like Kirsty that are looking at this data and saying, let me try to make sense of it. And if we're just sending it in with no information, that's going to be really hard, because it's like, where does Kirsty start looking in terms of what genes are relevant? Like, is this child nonverbal? Does this child have seizures? What is their intellectual level? Do they have an intellectual disability? Any phenotypic features, also in terms of do they have dysmorphic features, such as on their face? So there's so much in there. What specifically do you guys look for on those requisitions from people like me that are ordering testing in general, but my counterpart in peds, I guess? What do you guys look for on the test rec of like what's important? Like I just gave some stuff off the top of my head, but what else am I missing?
It's hard to say what specifics, because honestly, the more you can provide, the better. And that's tough because you only have so much time to fill out a requisition. And our requisitions at Blueprint, we ask for a lot of detail on purpose, right? Because there are concepts like variable expressivity and reduced penetrance and kind of just shooting in the dark and looking at what these variants tell us. I mean, Kirsty can, I'm sure, add more to this, but if we're seeing a change in an area that's really highly conserved, sure, that's suspicious, but wouldn't it be giving your patient the best possible chance of finding a quality answer related to their health? If I am to be specific about things, especially current health conditions of how the patient is right now, it really depends on the specialty that you're ordering from. So if it was like neurology and things like that, like developmental things like that are super helpful. And in pediatric space, it's something that we pay attention to. But we're also curious too of how did this happen in this individual? Is it new, or is it something that's inherited? So even if you have family member info of direct family members, that can be quite helpful for telling us what the next steps might be in terms of family variant testing. Kirsty, what do you want to add?
Yeah, Kirsty, I'm in there.
Yeah, I totally agree with all of that. I mean, from the interpretation point of view, more clinical information and more details is always better for us, because then that helps us to kind of interpret the variants more easily if we have more specific clinical information about the patient. And we know that's not always possible, though, so we can still work with kind of minimal clinical information. We can still do the interpretation, but it's easier and faster if we have kind of good, detailed, specific clinical information. And that's especially if we're doing a trio test, for instance, where we are comparing a patient's data with that of their parents, then it's really important to know if the parents are affected with the same disease or if they are healthy. So that kind of information really is helpful to us in the interpretation and helps, with the quality of the result that you're going to get.
And I think a theme throughout this episode so far has been the patient-first mindset. I know that's really important at Blueprint Genetics, where you guys are working. And how can healthcare providers also adopt this mindset so that, the healthcare provider and the genetic lab, if they're ordering from you guys, are on the same page? Obviously, we've highlighted a lot. Is there anything else to add to that part of the conversation?
I think that just traditionally in healthcare, patients are blindly trusting of providers in what's going to be ordered. So there is so much on the onus of the provider. And I think that we as the lab have to support the providers. We need to set the standard of quality of, for example, depth and coverage of what should be the minimum so that we're able to do these tests properly. But I also think it's empowering them to be able to access of what transparency should be, right? You should be able to know exactly how many genes are on there, how well they're covered, how much could this cost, or is there's pricing on there? There should always be a person they can reach to ask these questions, because if we as the lab are not empowering the providers, or at least giving them the tools to do it, I don't feel that we can do right by the patient then, because the provider is sort of our connection as the lab and industry to the patient at this point. So I feel we often have to work through the providers and empower and educate them.
Yeah, I think that's very well said, and that, we're trying to, all of us, achieve that patient-first mindset. And, a couple quick questions before we end. What is the turnaround time usually for tests that ordering providers should expect? And if it's longer than that, then they need to talk to the lab and be like, hey, what's going on? I think about panels, exomes, genomes. Is there number of, days, weeks in mind for you guys when it comes to this testing, or what Blueprint typically quotes?
Yeah, so when it comes to panels, we typically have a turnaround time of 4 weeks or less. And when it comes to whole exome sequencing, we're about 8 to 10 weeks. And of course, knowing being a GC myself and in the field, there are varying times for turnaround time. And ideally, we would love to be able to give results as fast as possible. And, over time, it is something with more resources we're able to do. But because Blueprint is so concerned about quality and making sure that we don't use up all resources and are able to offer a steady product over time, we are going to take some time to do these things. And that's because we are, exactly as Kirsty described, there are so many tiers that we want to go through to deliver that quality and the confidence, really, because quality is confidence, right? If I think a test is good quality, I'm expecting a certain tier of excellence each time. And that as a lab, we make sure we're offering everything out there and it's pretty clear what we're offering. So do I think there are certain patient scenarios that will require a faster turnaround time? Absolutely. And hopefully that is something we are able to offer in the future. But we are quite proud of the quality we deliver in the time that we're able to deliver it.
Yeah, no, that sounds like, what I hear from other labs in terms of turnaround time.
Right.
And if a patient receives results that, okay, we couldn't find a causative pathogenic variant that we think is leading to your symptoms, how often should they check back? I've asked a few guests this for longtime listeners of the show, but I just like to hear what people recommend for that, because I don't think there's necessarily a standard in the field for this. Unless there is, and I've just missed it.
No, I don't think it's missed. And I honestly, it depends like what lobe of my brain I'm using. If I'm using like the clinical lobe or the industry lobe, you know?
Yes.
And like clinical lobe, it depends how often you're seeing them, right? And they come to you if they've seen you and had this test and they're hoping for something different. I think Kirsty will definitely be able to add to it, but I think like minimum a year, right, for us to be able to see some of that. And if there is a change that happens before a year, Blueprint will reclassify variants and send out a new addended report to all providers who ordered. Yeah.
Not all labs do that.
Not all labs do that. Yeah. And at Blueprint, it's based off of if the variant's reclassified, perhaps because another individual added enough information that we were able to reclassify it. That's kind of how the stream goes.
Our database grew. There we go.
Right, exactly. And Blueprint, you know, is an international lab, so we're not just testing people here in the United States. We're actually getting a really diverse population. So we're really proud of that too. But it's tough, right? Because for reanalysis, West reanalysis, we offer that after a year, between 1 to 2 years. So it varies, and we just encourage, like, if somebody has a variant, even if it's from another lab, and they want to know more how Blueprint classifies it at this time, they're absolutely welcome to reach out to us to ask that, because we're happy to share if we've seen that variant before.
Yeah, that's a great tool for genetic counselors, other healthcare providers that order genetic tests, that if one lab is telling you, okay, we're classifying a variant as whatever, you know, either VUS, where we don't know, or pathogenic, benign, you can always check with other labs and say, how do you guys classify this one? You know, just take a poll, make sure that everyone's on the same page, and if there's a difference there, diving deeper. I wish we had even more time because you guys are lovely. So much great information. I wanted to give a shout out for a couple listener responses that we had on social media on what they think quality genetic testing is. Sandra said, can I get all 3 of affordable, fast, and accurate instead of just 2? I feel that one. That one is tough. Yeah. Lydia Siders, who was a previous guest on episode 98 years ago, said advice on what to do next. So certainly I know that that's something that is often mentioned in reports, saying, you know, a lot of it is we recommend genetic counseling, so that's kind of a standard I see in a lot of reports. But sometimes there is more specific, like genetic counselor and a parental karyotype or this or that. So sometimes that can be helpful. Also want to shout out Heidi and Haley. Holly, you both said the same thing, which I thought was so funny. I was like, did someone submit this twice by accident? Clinically actionable results, which, I mean, that's what we're getting at here, right? That's end of the day, that's what we want. So thank you so much to listeners that, you know, wanted to chime in for this episode, and make sure to follow us on social media at DNA Today Podcast so you can chime in on other episodes. But Kirsty, Rachel, thank you so much for sharing all your expertise on this. And I think it's great to have an episode where we dive into what seems to be kind of like an overarching idea of genetic testing, but you get into it, it really is a specific topic. So thank you both so much for coming on and being just lovely guests.
Thank you.
Thank you so much
for
having
pleasure.
us.
Thank you.
For more information about today's episode, visit DNApodcast.com, where you can also stream all episodes of the show. We encourage your questions, comments, guest pitches, and ideas. Send them all into info@DNApodcast.com. Search DNA Today on Twitter, Instagram, YouTube, Facebook, so you can connect with us there. And a favor, please rate and review the podcast on Apple, Spotify, or wherever you listen. This truly helps us climb the charts and allow more genetic nerds like yourself to find the show. DNA Today is hosted and produced by myself, Dineen. Our social media lead is Corinne Merlino. Our video lead is Amanda Andrioli. Thanks for listening, and join us next time to discover new advances in the world of genetics.
The genes of you and me, the genes of you and me are all made of DNA. We're all made of the same chemical DNA.